In this video, we will discuss about how this glycan has a dual role. That is, this glycan can help in the immune evasion. At the same time, these glycans can help in the immediate immune response. So these are different examples that are shown here. First one is about this bacterial colonization. In this case, the bacteria has the cell surface glycans, and these glycans are these glycans that are formed on the bacterial cell surface are similar to the glycans that are expressed on the host cells. So in this case, what will happen is that these glycans will prevent the host cell immune system from generating the immune response against this bacteria. At the same time, this bacterial the glycans that are expressed on the bacteria helps this bacteria to intravasate or entered into the host cells that can be seen here. So this is one of the way by which this glycans can help in the immune evasion of this bacteria, and also helps in this bacterial colonization, that is entering of this bacteria within the host cells. And the other way that can, and this is the glycans that are shown, that glycans with the surface bacteria with the surface glycans can enter the host. However, bacteria without the surface glycans will not be able to enter the host, as they don't have a medium to interact with the cell surface of the host cells. So in this case, a biofilm will be formed or generated by the immune system of the host cells, and later on this bacteria without the cell surface glycans will be taken care of by the immune system of the host cells. The other way that is shown is kind of a zoom in view of what has been happened in the part 1, that is immune evasion. How this immune evasion occurs is that when this bacteria has this modified PAM, this pathogen, this is something pathogen associated uh, some, I'm not able to recall its full form. So this has some modified PAMs or CPS. So what it will does is it has a similar glycan formations of the glycans that are formed by the host. So in this case, when the immune system interact with these glycans on the bacteria that are expressed on the cell surface of the bacteria, it will not generate immune response, and that helps in the immune evasion. Whereas when the glycans that are formed on the bacteria are different from the glycans that are expressed on the cell surface of the host, in this case the immune system receptors will detect these kind of glycans and generate an immune response. So these are known as PAMs. So these structures are known as PAMs. However, these modified PAMs or bacteria can modify itself to generate the glycans that are similar to the host. So in that case, there is immune evasion occurs, whereas in this case, the host immune response will be triggered. So, and the parts, another part is this host glycan utilization. So in this case, what will happen is that once the glycans enters the once the bacteria with the cell surface glycans similar to the host, or the bacteria with the cell surface glycans are able to enter the host. In that case, what will happen is when these glycans will use the host cell machinery and generate more replicates of this bacterial pathogen, and when they est- when they move into the extracellular matrix, they will use the host glycosylated proteins, and thereby that will help in the immune evasion. Whereas the so this is one of the examples that is shown here, that is toxin with the N glycans that are expressed on the host will be able to enter the host cells and replicates more toxins, and when these toxins will get move into the extracellular matrix, they can easily uh, when they move into the extracellular matrix, they will have these host glycosylated proteins. Thereby, their further replications will have more chances of immune evasion. The last thing is about the immune response. So in this case, what will happen is that in the part 4, when the bacteria, it is the elaboration of this part B on the right hand side. So in this case, what will happen is that when the bacteria has the glycan surface that are different from the host cells, then it will lead to the generation of this phagocytosis pro-inflammatory response. And so what will happen is it will activate this innate and adaptive immunity, and that will lead to the pathogen clearance. Whereas if this that will lead to the pathogen clearance, and what will happen in this phagocytosis and pro-inflammatory response is that this antibacterial autophagy will be generated, will be activated, and this will detect the glycans that are expressed on the bacteria, which are not similar to the glycans of the host, and it will generate the it will generate the antibacterial autophagy. That is, it will try to kill or clear off this bacteria with these pathogens. And during this case, both the innate and adaptive immunity will be activated. So in this case, the glycans can, this is some of the examples by which glycan can play a dual role. I hope you got an idea how these glycans can have a dual role. If you need to understand more about these glycans and its dual role, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.